The antibiotic testing you guys set up on Tuesday. Unfortunately, you guys tested a gram-positive organism and a gram-negative, and neither of the gram-negatives grew out very well. They were brand new cultures I'd just gotten from a company, and I think they were old cultures, and they didn't have time to grow out enough before we used them. So we're going to focus in on your gram-positive plate for our follow-up this afternoon. All right, so when we left off on Tuesday, we were starting to talk about modes of action or mechanisms of action of antimicrobial drugs. And this diagram is really antibacterial drugs because we've got a bacillus-shaped bacterium there. And it's just showing you different antibiotics and how they actually kill or inhibit bacteria from growing. And we're going to talk a little bit more about some of these. All right, so we've talked about before how a lot of the antibiotics, and again, traditionally that term means antibacterial. Many of the antibiotic drugs you take for bacterial infections work by inhibiting cell wall production. So we've talked about that before. Here's a diagram from another textbook, actually, which is just reviewing the cell wall. That's the peptidoglycan coating around the bacterial cells, and we learned before that is made from chains of 2 sugars that are abbreviated as NAG and NAM. You've got strips of those, and then those strips are held together through cross-links, and the cross-links are actually peptides, little short chains of amino acids. And so those NAG-NAM sugar strips are held together this way and then this way, so you wind up with a 3D peptidoglycan cell wall. All right, so these many of the antibiotics we take, they interfere with that cross-linking. So the NAG-NAM sugar strips still get made. The bacteria can still make those. Those are a type of polysaccharide. But they're just not getting linked together. So there's nothing to hold the cell wall together. It just falls apart as it's being made. There's a big group of antibiotic drugs. There's a big category called beta-lactams, and we'll come back to that shortly. Penicillin is one of these, and all of its relatives fall into that category. Cephalosporin, you may have heard of that one or seen that one before. This is how they work. They prevent the NAG-NAM sugar strips from being cross-linked. Remember, if you don't have a cell wall, bacteria, our body fluids are hypotonic for bacteria. So for most bacteria, so water flows into the bacterial cells. And without a cell wall, they tend to blow up. They burst. One thing about these is they only work on growing bacteria. So if you've got a live bacterial cell, but it already made its cell wall, the drugs aren't going to prevent it from making its cell wall because it's already finished its cell wall. So they actually won't be killed by these types of drugs. They only work against bacteria that are dividing and creating new bacterial cells in the process of making their cell walls. Now, they're still helpful because they knock out all of the young new bacteria, and then your immune system can swarm in and knock out the other ones that may still be around. All right, beta-lactam is an organic chemistry term. This is the chemical structure for penicillin. And remember when you see these geometric shapes, there are carbons at each of the points. They just don't draw them in to make the diagrams simpler. So you'd have a carbon there and one over here. My pen's not working too well. Here, 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 etc. And there are hydrogens attached to the carbons as well. They just don't show those either. But right in here in penicillin, they've highlighted in green there's a ring-shaped chemical structure. That's called a beta-lactam ring. It kind of looks like a square. So all of the antibiotic drugs in this category have that structure as part of the molecule. For example, here's another one that's in this category, methicillin. It's a semi-synthetic. What did we say that meant? It's a semi-synthetic. Yeah, it said they've taken, the chemists have taken a naturally occurring antibiotic like penicillin and they've altered it chemically. So like with methicillin here, it looks just like penicillin, but you've got an oxygen and a carbon and 3 hydrogens attached there and there. So the chemists did that in the laboratory and created methicillin. And again, why do they do that? What makes them better? A lot of times they're more stable in your stomach acid. They might be absorbed through your intestinal lining better so they can get into your bloodstream. They may be able to actually enter the bacteria, especially gram-negative bacteria. A lot of antibiotics can't get through their outer membranes, so that's another reason why they may work better as well. Or sometimes bacteria have enzymes that will destroy antibiotics. Like they might have enzymes that destroy penicillin, but maybe they don't destroy methicillin, so the methicillin can still work against those bacteria. All right, so among our antibacterials, you got a whole bunch that inhibit cell walls, then you got another big batch that work by preventing the bacteria from being able to build their proteins. So they interfere with translation. So we've got some translation diagrams here. They don't all work the same way. They can be divided up into different categories according to how they work. There are 3 big groups of antibiotics that inhibit bacteria from being able to build their proteins. And you may run across some of this terminology from time to time. Aminoglycosides, tetracyclines, macrolides. You'll especially run across aminoglycoside and macrolide antibiotics. Then among your aminoglycosides, some specific members of that group, streptomycin, gentamicin, neomycin, macrolides, erythromycin, azithromycin, that's your Z-Pak. Maybe you've heard of a Z-Pak before. Many of these are pretty strong, so if they can get inside the bacteria and you screw up protein synthesis, so they're going to work against lots and lots and lots of different types of bacteria. How do they actually do it? Some of your aminoglycosides, like this diagram is showing you, they attach to the 30S part of the ribosome, the small subunit, and they warp it. And that causes incorrect amino acids to be included in the protein. This is supposed to be the chain of amino acids up there at the top that's being built. So the wrong ones get incorporated. And then the tetracyclines and some of the aminoglycoside group, right here in green, that's supposed to be the drug. They attach and they block the next tRNA that's trying to bring in an amino acid. They stop that from happening. Then you've got some, so here this is supposed to be 2 amino acids getting bonded together. And chloramphenicol is an antibiotic that prevents that from happening. The macrolides attach to the ribosomes and they actually prevent, the ribosome is supposed to shift to the next codon, to the next one, to the next one, and they prevent that from happening. So that's how they work. So they don't all work in exactly the same way. But if you stop a bacterial cell from being able to build its proteins, it's going to die pretty quickly. Okay, then there are some drugs that we take that work by disrupting cell membranes, cytoplasmic membranes. Now, these are not all antibacterials. A good example is amphotericin B. It's an antifungal type drug. It's a pretty big molecule. That's supposed to be its chemical structure there. And what it does, this is supposed to be a fungus cell membrane. And you may have learned before that our cell membranes, the cell membranes of animals, have cholesterol as part of the cell membrane. And fungi don't have cholesterol. They have something called ergosterol that's very similar. It's a type of lipid that's present in their cell membrane. Well, this drug attaches to, it binds to ergosterol, and it creates pores in the cell membrane. So then the poor little fungi take up too much fluid and they wind up bursting. The problem with this drug is amphotericin B is really strongly attracted to ergosterol, but it's also slightly attracted to cholesterol. So that means the longer you take it, or if you take too much, like if you're somebody who thinks I should take 5 pills instead of 1 so I can get over my infection sooner, this drug can attach to your cell membranes and poke holes in your cell membranes. So you have to be concerned about that. All right, and then I think on Tuesday we talked a little bit about polymyxin. That's a drug that can poke holes in the cell membranes of many gram-negative bacteria. However, their cell membranes are very similar to ours, so they'll also poke holes in our cell membranes. So these are toxic. You can use them on the skin, but taken internally, they're pretty toxic. They're especially toxic to your kidney cells. You have all these delicate blood vessels in the kidneys that filter the blood and create the urine. And so those organs are especially sensitive to it. This is the one, I think on Tuesday we mentioned, unfortunately there are patients who develop infections of the blood with bacteria that are resistant to everything except polymyxin. And so then you're faced with this horrible choice. Am I going to die of my infection, or am I going to take polymyxin and probably destroy my kidney function, which that could kill you. And if not, if you survive it, you may wind up on dialysis for the rest of your life. So polymyxin is kind of a drug of last resort. All right, then there are drugs that inhibit other metabolic pathways, specific enzymes, very specific types of things. And, the idea is you need them to interfere with something in a microbe, not in a human. There is a drug called atovaquone. Eukaryotes, we don't have as many drugs available, but atovaquone is one that can be used with some protozoans and fungi, and it actually shuts down their electron transport chains. So they can't make ATP, they don't have energy, they don't survive. Then the sulfonamides, we actually talked about those on Tuesday. These were one of the early types of antibiotics as well, and they prevent bacteria from making folic acid. And all living things need folic acid in order to be able to replicate your DNA. So without folic acid, they can't do that, and the bacteria wind up dying. The way that one works, in bacteria take this molecule, which is abbreviated as PABA, or PABA, and that's probably a name that's like this long, some kind of organic chemistry name. They take that and they convert it to folic acid. So they have an enzyme or enzymes that do that. Thus, here, sulfamethoxazole is an example of a sulfonamide drug. So it looks like PABA. So the enzyme in bacteria that takes PABA and converts it to folic acid binds this drug instead. And that's an inhibitor. It inhibits the enzyme. It shuts those enzymes down. So they stop making folic acid. We don't make our own folic acid. We get it from our diet. And so that drug, if taken at appropriate doses, shouldn't be toxic for us. Okay, there's another group of antibacterial drugs called the quinolones and fluoroquinolones. And they inhibit an enzyme called DNA gyrase that bacteria use to help them with DNA replication. We've talked about how DNA is very compacted, really tightly inside a cell because it's so long. You got to really fold it up really tightly to make it fit in there. This enzyme helps unwind some of that tension so that the DNA can actually be copied. So the antibiotics in this group that's what they inhibit. If you can't replicate your DNA, you're not going to be able to create new bacterial cells. So probably the most familiar one in this group is ciprofloxacin, which is also known as Cipro. That's not unusual for that antibiotic to be prescribed for urinary tract infections. And then there are some newer ones in this group, gadafloxacin, gemafloxacin, etc., that work in much the same way. All right, then there are some other drugs that inhibit nucleic acid synthesis in another way, and they are referred to as nucleoside analogs. So these inhibit production of DNA or RNA. And a nucleotide is, remember, a nucleotide has three parts. You've got the nitrogen-containing base, that's your A, G, C, T, or U if it's RNA. Then you got a sugar, ribose or deoxyribose. And then you have a phosphate. That's called a nucleotide. A nucleoside just leaves off the phosphate, and your cells will attach the phosphate and make the full nucleotide, which can then be used. So the reason these are called nucleoside analogs is these are drugs that resemble, in the center here, you've got your A and your G and your C and your T, nucleosides. Those are the normal ones. These drugs resemble those, and they fool the enzymes that are being used for DNA and RNA production. And those enzymes use these drugs instead, and the drugs inhibit the enzyme. The enzymes shut down, and they can no longer continue to build DNA or RNA. And there are a lot of these. Many of these are antivirals. And some of them you may have heard of before. Acyclovir or ganciclovir. Has anybody ever heard of those before? Those are used for some DNA viruses. Sometimes they give kids with chicken pox one of those drugs to help shorten the amount of time the infection is going to take place. Herpes, chickenpox is a type of herpes virus. It can be given for shingles, any of the herpes virus type infections. Has anybody heard of this one before? AZT? Anybody know what that's used to treat? That's a HIV. Yeah, that was the first major drug that was used for HIV. AZT resembles T nucleotides, and HIV's reverse transcriptase that converts the RNA to the DNA will use this drug in place of T's, and then it gets shut down. It inhibits the enzyme. Somebody seen the Dallas Buyers Club, that movie? Yeah, that's where that's a story about a guy in Dallas who gets HIV back in the late 1980s. And so he goes on a quest to find and sell drugs to, this is really early during the HIV epidemic and didn't have a lot of drugs widely available yet. And so he's kind of creating a black market for HIV drugs for the many people who were already infected with HIV back during that time. So one of the ones they talk about a lot is AZT, because that was one of the first ones out on the market. And let's see, DDC up here, dideoxys. Where is it? Over here. DDC, dideoxycytidine. That's actually used as a anti-cancer drug. So cancer cells have to do a lot of DNA synthesis because they're dividing very rapidly. And so this drug will shut down their DNA replication. So that can limit the growth of cancer cells. However, guess what? It's also shutting down the DNA replication of your non-cancerous cells, which is why so many of these types of drugs that you take for cancer are very toxic, especially to not all of your cells divide, but like think about your bone marrow. You're constantly creating new red blood cells, new white blood cells, and these types of drugs will inhibit that. So that's why a lot of times cancer patients who are taking these drugs become anemic. They can't make enough red blood cells. They can't fight infections very well because they're not making enough white blood cells. Your hair falls out. You get sores in your mouth because you're not replacing your mucous membrane linings in your mouth as easily. We talked about drug spectrum a little bit on Tuesday as well. Remember, narrow spectrum drugs are drugs that act on a really narrow window or a number of different types of pathogens. Broad spectrum drugs tend to act against a wide array of pathogens. And this table is simply showing you different kinds of bacteria, different groups of bacteria, and families of drugs or types of drugs that they're susceptible to that will actually kill them. And notice this group over here, the mycobacteria. Good old Mycobacterium tuberculosis. It always shows up all throughout the whole class as a big pain in the butt type of bacteria. It's resistant to many things. And so what you see here is there is a drug called isoniazid that can be used in tuberculosis patients. And streptomycin can be used in some cases. There's another one, tobramycin, that can work against some strains of TB, but it's pretty limited. We don't have a lot of drugs that work against Mycobacterium tuberculosis. Remember that waxy cell wall? They have that special barrier, and that's preventing entry of most of the drugs that we do have. Another thing on here as well, in general, we have more different groups or families of drugs to treat gram-positive infections than gram-negative, and that's that outer membrane of the gram-negatives tends to prevent some types of drugs from getting in. There are a couple of other groups of bacteria, the chlamydias and the rickettsias. These are little teeny tiny bacteria, and many of them, now they are bacteria, they're not viruses, but many of them really need to go inside a host cell, almost kind of like a virus, and live inside a host cell and have that host cell support them. So many of these are intracellular parasites. Really teeny tiny, and we don't have a lot of good drugs to treat them. A good example of a rickettsial infection is Rocky Mountain spotted fever. A lot of those are delivered by ticks to people. And I imagine we've all heard of chlamydia before. That's the most common sexually transmitted disease. And we do have drugs to treat chlamydia. Penicillin combats a lot of the sexually transmitted disease forms of chlamydia. And then keep in mind, as we've talked about before, if you take a broad spectrum antibiotic, you wipe out a lot of your own normal microbes, that just opens the door for other things to grow. So you can wind up with an infection that you didn't have before. This is a table from your textbook that is showing you how we don't have a lot of antiviral drugs yet. We're beginning to develop more of an arsenal, but so far we don't have very many of them. But it is showing you, well, how do antiviral drugs work? There are a few examples of drugs that inhibit a virus from entering its host cell. Many of them inhibit DNA or RNA synthesis. And there are a few that will actually prevent the virus from assembling itself or even getting out of its host cell. And if you look closely at these, the effects of the drug, what are they actually doing? And many of these listed are HIV, anti-HIV drugs. So we really have the most of our antiviral drugs work against HIV, the ones that we do have. And that's because there's been so much intense research into HIV. We've come up with a lot more drugs for it than anything else. Also, its odd life cycle. It does that RNA to DNA, that reverse transcription. And so we're kind of lucky that HIV fell into that category, because that's another reason why we can come up with a number of different kinds of drugs to impact HIV when we haven't had such good luck with many of the other viruses that are out there. All right, efficacy tests. Does a drug have efficacy against a microbe? That means, does it work? Is it going to kill or inhibit the microbe? What is its efficacy? And this is what you guys, if these had worked out ideally, this is what you guys would come back and see today. You would have your agar plate with a lawn of bacteria all the way across, and you've got your antibiotic discs in the center. And then, as you guys are going to see after the break, you actually measure. This is called the zone of inhibition. You take a ruler and you measure across that the diameter of that circle, that clear zone that you see there. And then you look on a chart, and the chart tells you, okay, based on the size of that zone, will that antibiotic actually work? Would it have efficacy in a patient? Like this one, you've got a little teeny tiny clear zone around the disc. So that means, well, yeah, that drug will kill the bacteria or inhibit them from growing, but only at a really high concentration. So can you achieve that concentration safely in a patient? Probably not if you have that little tiny zone. If you have a broader zone, the drugs are diffusing and getting lower and lower in concentration as you get further and further away from the disc. So if you have a big zone, that's usually an indication, well, this drug still works at a low concentration. That's probably going to be a good one to use in a patient. But you actually have to measure them and compare them to a chart that tells you, okay, if it's 12 millimeters, it'll work. If it's 10 millimeters, it won't work. And that's traditionally how these were done. This is going away in a lot of clinical laboratories, and now a lot of it's more automated. They grow little teeny tiny liquid cultures of bacteria with many different types of antibiotics, not just 8. And if the bacteria grow or if the bacteria don't grow in the presence of a drug, a computer will scan it and figure that out and let you know which drugs are likely to work and which ones are not. Okay, routes of, as you guys know, different drugs have different routes of administration. Usually we prefer to take things orally, but that doesn't always work for some types of drugs. Some drugs will get destroyed in your stomach acid, or you can't absorb them properly through the intestinal lining. And also the route of administration affects the level of the drug inside the body. So over here on these graphs, on the y-axis, that's the concentration of the drug in the body. This is over time. So when you take a drug orally, you got a little lag. Because you got to swallow the pill, your digestive system has to break the pill down, you've got to absorb the drug through the intestinal lining and into the bloodstream. Then the levels go up, your liver starts breaking it down, you start urinating out the drug, and the levels go back down. So then you got to swallow another pill. IM is an intramuscular injection. So when that happens, you have an immediate big-time rise in the drug concentration because your little tiny blood vessels in the muscles, the capillaries, will absorb that drug really quickly. So you'll have a big burst in concentration, and then it will drop back down. And you have to have another shot or if you need one. Intravenous IV, that's where they're putting your drug in a bag of IV fluids. And so as long as you're hooked up to the IV, you can keep your drug level up here at a high level. So if somebody has a really serious infection and you can't tolerate these ups and downs with the drug concentration, you got to keep it high to try to fight the microbes off, then you might need an intravenous antibiotic. This is important too. When you guys take Biology 201, you may have talked about the blood-brain barrier. The capillaries in the brain and in the spinal cord don't let as many things pass across as the capillaries in other parts of the body. There you have a more tight seal around those capillaries. So that's important when it comes to drugs. There are a number of drugs that can't get out of the blood and into the brain or the spinal cord. They don't cross, they call that crossing the blood-brain barrier. So that winds up being an issue sometimes if you have an infection of the central nervous system, the brain or the spinal cord. That's true if you have brain cancer as well. A lot of cancer drugs don't get across the blood-brain barrier. So they may have to give you a drug that opens those blood vessels up and makes them leakier so you can actually get the drug into the brain where the cancer is occurring. All right, drug safety and side effects. Drug safety and side effects, very important as well. Now, we all know there can be a lot of side effects with drugs because we all see the commercials where most of the commercial is reading off a laundry list at like 90 miles an hour of every horrible thing that will probably happen to you if you take these drugs. So a lot of that Food and Drug Administration, a lot of that's legal, done for legal reasons. And the Food and Drug Administration requires a lot of that. So, when they're testing drugs in people before they bring them to market, they have to survey the people who are part of the trial. And, if they report anything, that may have absolutely nothing to do with the drug. But, if they have a headache, they have to report it. Might not be caused by the drug, but it still has to be reported. And if they notice, a few people who took this drug had headaches, then headaches has to be listed as a possible side effect. So some of them probably really are true side effects, and some of them probably are not. They were just things that were reported when the drug was being tested and may not have anything to do with the drug. The side effects can be toxicity. That's where the drug is actually toxic for some part of your own body. Allergies, like penicillin allergies, that's where your immune system thinks the drug is a foreign invader, like a bacterium or a virus, and tries to attack it and overreacts to it. And then disrupting your good old normal microbes. We've talked about that many times and how that's not good for you. And why are these drugs toxic? Because when you develop a drug, it's supposed to be something that's only going to screw up the microbe, not going to screw up things in our own body. So sometimes the mechanisms are not well understood. Yes, some drugs do a great job at disrupting something going on in a microbe, but they're also at a lower level doing things that can be damaging to our own bodies. And sometimes we don't even know specifically what those are. A lot of times these problems occur in the kidney and the liver because they filter drugs out of the blood. And then the nervous system tends to be really sensitive to drugs as well. Because if you start killing your neurons don't get replaced. So the neurons in your nervous system are pretty sensitive to many drugs. Plus they're very active cells, very metabolically active. And then we just talked about polymyxin. And some of the antibiotics in this group, the aminoglycosides, can have fatal toxic effects in the kidneys, which are not so good. All right, and then there are some toxic effects that are really just cosmetic. So there is a drug, an anti-protozoan drug, called metronidazole. So you might take that if you had a Giardia infection, a protozoan. And it causes a black hairy tongue. And most people don't want to have a black hairy tongue. So that's kind of a disturbing side effect. And I finally remembered last semester to look up what is causing the black hairy tongue. And what it is, the little bumps on your tongue, those are called the papillae on your tongue. This drug causes those to increase in size. And then those larger bumps on your tongue start collecting dust and food particles and things like that, and that turns black. So you don't really have a black hairy tongue, you just got a lot of gunk on your tongue that's being trapped if you take this particular drug. But I guess it looks pretty gross, so most people don't want to have a black hairy tongue. Yes, it does go away later. So try not to get a protozoan infection because you might wind up with a black hairy tongue. And then tetracyclines. Tetracycline is not used all that much anymore. It used to be used a lot, like back in the 60s and the 70s, and it can discolor the teeth. It can cause the enamel to turn yellow. My brother actually has some tooth enamel damage from taking tetracycline as a kid. Yeah, drug safety and side effects. Sometimes drugs can trigger anaphylactic shock. That's a huge, over-aggressive allergic reaction, which can cause your body to go into shock and can actually kill you. Because what happens to your blood vessels during inflammation? If you have inflammation around a wound, you have vasodilation, your blood vessels dilate. That's to increase the blood flow. into the area where inflammation is occurring, because you need to let all the good white blood cells and other things that are going to help fight infections and clean up damage, they need to get out of the blood and into the surrounding tissues. Are you looking for your textbook? It's up in my office. Yeah. Go ask Miss Hutchison up there. She can go into my office, and your book is on my desk. Okay, so if you have anaphylactic shock, what's happening is you're having... he does, yeah. Oh, that's right. He came in before he was looking for his keys, yeah. He found his keys, too. Okay, so if you go into anaphylactic shock, that's where you're having an allergic reaction across the whole body at one time. So that means your blood vessels dilate across the whole body at one time. So guess what happens to your blood pressure? If your blood vessels, your blood pressure goes way down. It plummets. And it can drop so low it can actually kill you. It can slow down your blood circulation. You're not circulating blood anymore to your vital organs. All right, then we've talked about your normal microbes many times, and how if you take a strong drug, knocks out a lot of your normal microbes, it opens the door for other things to grow. Like Candida albicans yeast can overgrow, C. diff can grow and cause disease as well. When that happens, there's a term we have not used before, super infection. And that doesn't mean like a really strong, powerful infection. It just means an infection on top of another one. That's called a super infection, an extra infection that you're getting because of the treatment that you have received. So these things are big-time problems in patients who are undergoing medical treatments, because you got all these nasty microbes that are in those types of places in large numbers, because you got a lot of sick people, and then sick people have weak immunity. And that's why those are hospital or healthcare acquired infections, and they're a big-time problem. One of our faculty members, his wife has pancreatic cancer. She almost died a couple of weeks ago from a healthcare acquired infection. So it's very common. About 100,000 patients a year in the United States die from healthcare acquired infections. So things they did not have when they entered the hospital. Okay, then drug resistance. We've been talking about this quite a bit as well. This is where our genetics comes in. Microbes are good at mutating. The mutations we've talked about, that can lead to drug resistance. Horizontal gene transfer, that can lead to drug resistance. Things like transformation and conjugation, those types of things that we talked about earlier in the unit. Microbes are good at changing. And you know how this happens, so this is supposed to be mimicking this occurring in a culture, like in a liquid culture. But think about this happening in your own body as well. So what they're showing you there on that Petri dish in white, those are bacteria that are sensitive to a drug. So the drug will actually kill them. And if you have an infection and you've got billions of these bacteria in your body, within that population, you're probably going to have a few that are at least partially resistant to the drug. The drug's not having as much of an impact on them. That's what you're seeing there in orange. All right, and so what happens is if you start treating those bacteria with the drug, the sensitive ones die, the resistant ones don't. And so if you keep exposing your body or a culture to this drug and keep growing it, those resistant ones take over the population. They keep multiplying and growing. And then if this is happening in the body, then you can transmit those to other people, and now they've picked up the drug-resistant microorganisms, whatever they may be. So this is an example of a natural selection where you have created living things adapt to their environments. The strongest survive or get selected for in particular environments. That's what drives evolution, changes of living things. And microbes can evolve very, 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 very quickly because they divide very frequently. So this is happening all the time. It's a big-time problem. But a couple of terms that are becoming more and more and more buzzwords. Probiotics. We hear about foods with probiotics all the time. People drinking smoothies with probiotics. That means you're consuming live microbes like in yogurt and other types of products that are supposed to colonize your intestinal lining and increase your numbers of friendly microbes along your intestinal lining. Those are probiotics. Prebiotics, that term is referring to nutrients that you're consuming, foods and so forth, that encourage the growth of good, beneficial microbes. So prebiotics, you're not actually consuming the microbes themselves. You're just trying to encourage the growth of the good ones. So there's a lot of intense research into this area. You have to be careful, though, because there are a lot of kind of kooky websites out there with information about probiotics and prebiotics that's not really based on any kind of scientific study. And so you have to be kind of careful with that. But there is a lot of research being done in this area. So I think down the road, it's being discovered that when you look at the normal microbes of people who are diabetic versus not diabetic, or who have other types of like Crohn's disease versus people who don't have Crohn's disease. Even when you get down into like socioeconomic things, like people who are living in poverty versus people who are not living in poverty, people who eat a vegetarian diet versus people who don't eat a vegetarian diet, people who eat a lot of local produce versus people who don't. You find different populations of microbes living in their intestinal tract. So there's a lot of interest in trying to figure out, well, is that impacting our health? Yes or no? You know, there are people who think the microbes living in your intestinal tract can play a big role in whether or not you develop type 2 diabetes or not. And if so, can we replace or give people who have type 2 diabetes the proper mixture of microbes that should be living in their intestinal tracts? Will that help them? Will that help reduce the symptoms of things like type 2 diabetes? There's going to be all kinds of interesting things coming down the road in that area. Okay, when we say microbes are resistant to drugs, how? You know, why do the drugs not affect them? And here we're mainly focusing in on bacteria. Again, a lot of bacteria just have natural resistance to particular drugs, and that could just be due to their structure, gram-negative versus gram-positive. You got that outer membrane, yes or no? Or if you're a mycobacteria, do you have that waxy cell wall that blocks a lot of things from getting in? Some things lack the target of the drug, like if the drug impacts a very specific enzyme, if you don't have the enzyme, the drug's not going to do anything. Some bacteria don't transport the drugs in. Remember like the citrate, when we did the citrate test, and the citrate users were able to transport the citrate out of the agar into the cell. The citrate non-users couldn't do that. That's why they weren't able to grow. Same thing with drugs. Drugs, many of them have to move through some kind of a membrane protein to get into they have to hijack a membrane protein to get in. So if the bacteria don't have those membrane proteins, they're not going to be able to get into the bacteria and do nasty things to them. And then over here, in gray there, that's a membrane protein called an efflux pump. Those are pump proteins. What they do is they pump out wastes, but sometimes those pump proteins will pump out drugs. So the drug might make its way into the cell, but then it just gets pumped right back out. Then it's not going to be too effective. So sometimes bacteria have those as well. That's an issue in cancer. You know, cancer cells can become resistant to drugs the longer you take a particular drug for cancer treatment. And our cells have efflux pumps as well. So sometimes the cancer cells change and start pumping the drug out when they weren't pumping it out before. So that doesn't apply to just bacteria. Okay, that's natural resistance. Acquired resistance, you've got an organism that wasn't resistant to an antibiotic drug, and it becomes resistant to it. And that could be due to a DNA mutation of some kind. or you pick up drug resistance genes through one of those horizontal gene transfer methods we talked about, like conjugation, transformation, and transduction. They're picking up genes that they didn't have before. A lot of times these genes are on plasmids, like they're showing you there. It's not unusual for those genes to code for some kind of an enzyme that breaks down the antibiotic. And so if you wind up picking up that gene when you didn't have it before, now you're going to make the enzyme, or the bacterial cell is going to make the enzyme and break down the antibiotic. So these are some various methods by which bacteria can resist drugs. Okay, a couple things I want you guys to be aware of, because I think you're going to hear some of these things mentioned in healthcare settings when you're dealing with patients with infections. One is ESBLs. These are an emerging problem. What ESBL means, extended spectrum beta-lactamases. So beta-lactamases, so you have beta-lactam antibiotics. Those are like penicillin, methicillin, ampicillin, amoxicillin. There are tons and tons of those. That's a big group of antibiotics. A beta-lactamase is an enzyme that destroys a beta-lactam antibiotic. So the bacteria make those. Some strains of bacteria make these enzymes. And there are a lot of different versions of those enzymes. So like some of them might destroy penicillin, but not anything else. Maybe it'll destroy penicillin, but it doesn't destroy methicillin or amoxicillin, something like that. What's happening, though, is we now have bacteria that are making extended spectrum beta-lactamases. These are enzymes that destroy essentially every kind of beta-lactam antibiotic. So you're knocking out a whole family of antibiotics that could be used to treat infections. So this is a growing problem because there are a whole bunch of microbes that cause infections, especially healthcare-acquired infections, that traditionally have been treated with beta-lactams. That's what you use to treat these infections. And now we have strains of these things that are making these enzymes that destroy all those antibiotics, so they don't work anymore. So there are ESBL-producing strains of Klebsiella pneumoniae, Klebsiella oxytoca, E. coli, Salmonella, Proteus, Pseudomonas. These are among the organisms you guys had for your unknown project. So these are all common environmental or intestinal bacteria. When they get into other parts of the body where they're not supposed to be, they can cause pretty serious infections. I didn't give any of you guys the ESBL types, though, so don't worry. You weren't working with the kinds that make these nasty enzymes. But just to kind of illustrate how this is a growing problem, these are two maps of Europe. This is actually from 2006, so this is 8 years ago. The problem is no doubt getting worse. But over here, this is a map for E. coli. This is for Klebsiella pneumoniae. And what this map is showing, when E. coli is isolated from patients with infections, or when Klebsiella pneumoniae is isolated from patients with infection, what percentage are the ESBL-producing types that are resistant to almost all of the beta-lactam antibiotics? So the darker the color on the map means a higher percentage of these bacteria are making these ESBLs. And this is from 2006, so we're 8 years later. So I imagine it's gotten worse since then. I need to find an updated map and compare the two and see how that appears. If you notice, it looks like it's a bigger problem over in Eastern Europe. So that's the poorer part of Europe. You probably don't have healthcare, and hygiene practices are probably not quite what they are in the western parts of Europe, so that may partially explain why you have more of these problems over in those areas. A lot of these things, these really highly resistant strains of bacteria are appearing in Asia first, like in the really heavily populated cities in India. India is a place where a lot of these things are emerging first. But they're spreading from there to other parts of the world. It's getting kind of scary. I think I was telling y'all not too long ago that there are strains of gonorrhea now that are resistant to almost every antibiotic we have. That's pretty scary because gonorrhea is pretty common. And typically you just took an antibiotic and it was gone. Now there are people who are dying from gonorrhea. Oh, another couple of other things too. With why we're having increasing problem. I knew there was something else I was trying to think about. There are, especially when you get into poor parts of the world, there's no regulation on the use of antibiotics. So you can go to any store and buy whatever antibiotic you want if it's available, and take as much of it as you want for however long you want to take it. There's no regulation, no control. So you've got a lot of people who are just kind of routinely taking these antibiotics. The more these antibiotics get used, the more likely it is that the bugs are going to mutate and become resistant and spread around. And then agriculture. Have you guys heard about agricultural use of antibiotics? This is a huge problem as well. In fact, some people think the agricultural use of antibiotics is largely to blame for the resistance problems we're having now. But you get into a lot of farmers just routinely give their cattle and pigs and chickens and things like that antibiotics as a prophylactic. What did that mean? Prophylactic? Preventative. Yeah, if you're giving somebody a drug prophylactically, they don't have an infection, you're just trying to prevent them from getting one. So you've got all this livestock and chickens and so forth that are being given these antibiotics for their entire lives to try to prevent infection and allow them to stay healthy and get big and beefy and bulked up so that they're ready for the meat market. So a lot of people feel that's where a lot of our antibiotic-resistant bacteria are coming from. Remember we talked about MRSA, methicillin-resistant Staphylococcus aureus. This is a nasty strain of Staphylococcus aureus. Some people think that came from pigs who were being given methicillin as a preventative type antibiotic. There's a school of thought that that's where the MRSA bacteria came from that are such a big time problem these days. All right, so what are we going to do? How do we combat drug resistance? How do we get around this? Well, we have to come up with new drugs. It's expensive for pharmaceutical companies to come up with new antibiotic drugs. It's a long, expensive process. They're working on it, so hopefully we will have some things down the road. A lot of times those aren't very profitable drugs because people aren't willing to pay a whole lot for them sometimes. So drug companies would rather work on other types of drugs that they can make more money from, they can make more profit from as well. Also, knowing what you're infected with. If you have a viral infection, a physician does not need to prescribe you an antibiotic drug like penicillin or ampicillin or any of those types of things. They don't work against viruses. A lot of patients don't like that. When you go to the doctor and you're feeling lousy, you want to leave with some drugs. You don't want to be told, well, you got a virus, you're just going to have to kind of ride it out, take some Tylenol. So I think that might have played a role in why a lot of times patients were being prescribed antibiotics, even if the physician felt that they had a viral infection. That's not the only thing, though. So it's not unusual for if you have a viral infection, you can have a bacterial infection that follows, especially like up in the sinuses. You can have a viral sinus infection, and then a few days later, you start feeling worse, really high fever. That's when you develop a bacterial sinus infection on top of the viral one. And so a lot of times physicians were going ahead and giving you an antibiotic prophylactically. Let's prevent you from getting a bacterial infection on top of the viral one. But we really can't keep doing that type of thing with resistance that's emerging. And then on top of that, you've got, you need to they always tell you, finish your full course of antibiotics. If it's a 10-day course, take the whole course, even if you feel better after 2 days. A lot of times people don't do that. If you don't, though, what can happen Like we were saying, if you had a bacterial infection, you've probably got trillions of these bacteria inside you, or billions at least. Within that population, you've got some in there that are at least partially resistant to the drug. They're not as sensitive to the drug. So you might be feeling better because the drug after 2 days has knocked out 99.9% of the bacteria that were making you sick. But if you still got that 0.1% left and your immune system doesn't wipe them out, and now they're going to overgrow, they're going to overpopulate, and now you've got this big population of bacteria that are at least partially resistant to the drug. And then those might get spread to somebody else, and they might do the same thing. And over time, it's believed that's one of the ways that antibiotic resistance is spreading as well. All right, so you guys will be hearing a lot about antibiotic resistance during your careers. It's going to be an increasing problem. It's pretty scary when you think